So in this video, we're going to talk about the structure of the nephron. And the nephron, remember, is the functional unit of kidney filtration. And you find about 1 million of these in each kidney. Now, each nephron has several key parts. It's got a renal corpuscle, which is the first part. That's where filtration occurs. That leads into a proximal convoluted tubule. So as a tube, it has cells that have a wall, and there's a fluid-filled center. So the proximal convoluted tubule. That leads into a nephron loop that dives down deep into the medulla of the kidney, and it comes back up and then turns into a distal convoluted tubule that then leads into what's called the collecting duct. But these are the main parts of the nephron. So renal corpuscle, proximal convoluted tubule, or PCT, nephron loop, and then distal convoluted tubule, which is the DCT. And there's actually two major types of nephrons in human kidneys. We've got cortical nephrons and juxtamedullary nephrons. The difference between these two nephron types, you guys, is where their nephron loop is primarily located. Okay. Now, in both cortical and juxtamedullary nephrons, the proximal and distal convoluted tubules are in the cortex. That makes sense. But what's different about the cortical versus the juxtamedullary nephrons is that cortical nephrons have a very short nephron loop. Like their nephron loop does not dive down very deep into the medulla. It's shorter. That's significant because the nephron loop's involved with concentrating urine. So if the nephron loop's shorter in a cortical nephron, that means it's not so good at forming highly concentrated urine. That differs from the juxtamedullary nephron because their nephron loops extend deep into the medulla. They go down way deep in the medulla, and it's a big old long loop that goes in the medulla. And the function of this long nephron loop then is to form highly concentrated filtrate, which then becomes highly concentrated urine. So let's say if you guys were in a desert, which one would you want to use more? The cortical nephron or the juxtamedullary nephron? Juxtamedullary, right? If it's concentrating your urine, that means you're retaining water. Like you're not losing water into urine. You're actually reabsorbing it. So that if you're in a desert, you want to use more of these juxtamedullary nephrons. The idea being that you'd hold onto your water better than if you only had cortical nephrons. However, in humans, most of our nephrons are the cortical variety, not the juxtamedullary variety. So that we do a pretty good job of holding onto our body's water, but we still make pretty watery urine. This differs from other like desert mammals who, they're adapted to the desert and they need to be able to retain their water really well because water is a scarce commodity there, right? So that like some desert mammals produce urine that's like a paste. Like the desert rat will produce pasty urine rather than watery urine because they do such a good job retaining their water that what comes out isn't even really watery, it's more pasty. And so it's just like a thick kind of crystalline yellow paste instead of any kind of fluid, watery, type of urine. So they would have a lot more of the juxtamedullary variety of nephrons, right? We're stuck with the cortical variety. We do a pretty good job of holding on to water, but we still produce watery urine. Now, this shows the two types of neurons, you guys. You can see that between the cortical and juxtamedullary, you can see that most of the nephron is found in the cortex, and in both varieties. So you got the renal corpuscles are both in the cortex. The proximal convoluted tubules are both in the cortex. What differs, though, is the nephron loop is shorter in a cortical nephron, and the nephron loop is much longer in a juxtamedullary nephron. And the difference then between these is their ability to form concentrated urine. So if it's a longer nephron loop, there's more opportunity to reabsorb water and solutes. So it's actually easier to make, more highly concentrated urine. And remember there's four main parts to a nephron. So it starts with the renal corpuscle, which is here. That leads into a proximal convoluted tubule. It's convoluted because it's all over the place, right? Mm-hmm. That eventually leads into a nephron loop where we have a descending limb and an ascending limb, okay? And then that leads into a distal convoluted tubule here in the back of the cortex. The distal convoluted tubule actually associates with the corpuscle at a place called the juxtamedullary apparatus, or juxtaglomerular apparatus. We'll talk about what that means and what it is in a little bit. But the distal convoluted tubule eventually drains into the collecting duct. Now, technically, the collecting duct isn't part of the nephron, but it is the next segment of this whole pathway of tubules. And basically drains that fluid into the collecting duct. Now, the collecting duct can still modify that fluid further. So it's not technically urine yet. What's inside these tubules is actually still called tubular fluid or filtrate. And it's not until the end of this collecting duct, way down deep in the medulla, where the collecting duct meets at something called the papillary duct, which drains fluid across the papilla into the minor calyx. Once in the minor calyx, that fluid's now is what it is. You can't modify it any further once it's a minor calyx. But in the collecting duct, yes. Distal convoluted tubule, absolutely. Nephron loop, definitely. Same with the proximal convoluted tubule. All that can all modify urine, so you can actually reabsorb or secrete substances along this pathway. So what's the first part called again, you guys? Renal corpuscle. That leads into a proximal convoluted tubule. It's winding around, right? And then that leads into a descending limb of the nephron loop, and then an ascending limb of the nephron loop, and then our distal convoluted tubule. Remember, see how the distal convoluted tubule comes back and reassociates with the renal corpuscle? That's going to be important, you guys, because the end of the nephron associates with the beginning of the nephron. And it's in a way for the nephron to check what it did. It kind of can see how it modified that fluid. Yeah, it can see if it did its job right. And it can change then what it's doing to make sure it's doing the right thing. So it's pretty cool. How it can kind of see what it did near the end of the nephron. And the association here forms the juxtaglomerular complex near the renal corpuscle and distal convoluted tubule. Now, there's several key mechanisms of urine formation, and nephrons possess 3 of these mechanisms to make urine. The first of which is filtration. Remember, filtration is the process of, like it sounds, filtering or forcing fluid across the semi-permeable membrane. The membrane that's involved with filtration is the glomerular membrane. So at the glomerulus, which is in the renal corpuscle, remember that's where blood vessels are actually kind of coming in and intertwining there. We get our filtration membrane there, and that's where this occurs, filtration, you guys. And filtration only occurs at the glomerulus. So what gets filtered then are smaller molecules like water, ions, wastes, amino acids, certain gases. Those can all be acids and bases. Those can be filtered because they're small enough. Larger things like cells, so red blood cells, white blood cells, platelets, and large proteins like albumin, or even immunoglobulins, those can't cross the filtration membrane. They stay in the bloodstream, okay, under normal circumstances. So what gets filtered are smaller things. So think of like salty water. That's what gets filtered. Now, beyond that, then what we have then is something called tubular reabsorption and tubular secretion. Tubular reabsorption means that you actually can move substances from the tubular fluid back into your tissue fluid, which can then get reabsorbed back in your bloodstream. Okay? So reabsorption is the process of moving fluid from the inside of the nephron back into your interstitial fluid of your kidney, which then can get reabsorbed back into your bloodstream. Okay? So what do you guys think gets reabsorbed? What might you want to reabsorb from your nephron? Yeah, sodium. Like you don't want to lose all your sodium in urine. Yeah, maybe some smaller amino acids. Water, absolutely. You don't want to lose all your water in urine too. So some of that gets reabsorbed back into your interstitial fluid, which then get reabsorbed in your bloodstream. What about like ammonia and different wastes? Do those get reabsorbed? Nah, exactly. So reabsorption is a selective process, and it involves transport proteins. So actually, little transport proteins actually can use ATP or just facilitate diffusion of molecules across the tubular membrane. And that's reabsorption. And secretion is the opposite process. So tubular secretion is where you take substances from your bloodstream and interstitial fluid and then deliberately put those into the nephron. What do you guys think you might want to secrete into the nephron? Yeah, toxins, absolutely. And so with toxins, then that includes certain drugs, especially things like penicillin and other drugs. Those get secreted. Your body treats them as a toxin. Like your body doesn't differentiate between legal versus non-legal drugs and toxins. You know what I mean? Like it treats them the same way. But yes, like those can get secreted because you want to eliminate those from your body more rapidly. Would you want to secrete red blood cells? No, you want to. Yeah, that would make sense. What about, would you want to secrete glucose into your nephron?
Yes.
No, because it's a nutrient. Yeah, you want to keep it. It's a nutrient. You want to hold on to it. Even if your blood glucose is too high, your kidneys still don't secrete glucose.
Aren't there some medications out there that like force your kidneys to excrete?
Yeah, you raise a good point. So the way you can actually, let's say there's certain medications that can actually change how the way you make urine. And what those medications would do then was affect any of these processes. So, for instance, let's say if you wanted to have someone urinate out more acid, instead of having them reabsorb that acid, then you can block tubular reabsorption of an acid. And then by blocking reabsorption, then you can kind of ensure that it's going to end up in urine. Or let's say if you wanted to make sure that someone can not secrete a certain drug to a certain extent, you can give them another drug that can block tubular secretion of that drug, and that can prevent them from eliminating too quickly in their kidneys. So there's just different mechanisms you can change here with the urine formation. But there's only 3 of these mechanisms. You got filtration, tubular reabsorption, and secretion. Where does filtration occur? Yeah, and only the glomerulus. And it occurs there because the glomerular capillaries are thin. But whereas reabsorption and secretion occur all along the length of the nephron. So it's going to occur along the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. Now, it turns out, you guys, that we can actually filter our entire blood volume 60 times a day. So if your blood volume is like 5 to 6 liters, you're talking about quite a bit of volume of blood every time each day. And our kidneys use about a quarter of our oxygen supply at rest. It takes a tremendous amount of energy to make urine. We kind of take it for granted. We just say, oh yeah, we made urine, whatever. It's a waste. But it actually takes a lot of energy to even make that. It takes energy to eliminate those substances from your body. And it's an investment. Although it takes energy to eliminate those wastes, the alternative is worse, right? So it's kind of interesting. And what's also really cool too is that although we can filter our blood 60 times a day, I don't mean like any segment of blood, our entire blood volume 60 times each day, what we make is far less than, something like 700 liters of blood. That's not even possible. We don't have much fluid in our body. Instead, what you end up making is something that's a very small percentage of what was originally filtered. That's what you actually urinate out. So if you think about maybe if you only urinate out like a couple cups of urine a day because you were dehydrated, that's not because you didn't filter a lot. That just means you reabsorbed a lot of that fluid. And what you created, bless you, was a very highly concentrated fluid. So it's actually really, really neat. And, depending on, it'll look differently depending on its concentrations. If it's more concentrated, it'll be darker. If it's less concentrated, it'll be less dark. We'll talk about that later. But what this slide shows you guys are these 3 main mechanisms along the nephron. To just kind of orient ourselves what we're looking at here, we got our glomerular corpuscle, or capsule, right? So our glomerular capsule, our proximal convoluted tubule, our nephron loop, distal convoluted tubule, and then the collecting ducts. Now, in green, this is showing filtration. And the only place filtration occurs in the kidneys is at the level of the glomerular capsule, because this is actually going to occur at the glomerular capillaries. And we'll talk about how that works here in a little bit. Now, in the proximal convoluted tubule, you can get both reabsorption and secretion. We can tell that blue or purple here is reabsorption because you're removing substances from the inside of the tubule back into your bloodstream. That's reabsorption. So what are some things you might want to reabsorb? Yeah, water, absolutely. Sodium, other ions, potassium, right? Chloride, things your body needs. And you might want to secrete different things like certain wastes and drugs. That occurs in the proximal convoluted tubule. In fact, most reabsorption occurs immediately in the proximal convoluted tubule. Like 90% of the sodium that was filtered, you guys, gets reabsorbed right away in the proximal convoluted tubule. So what allows the PCT to reabsorb things really well is that the cells that line the inside of the proximal convoluted tubule, they're simple cuboidal, but they have lots of microvilli. And those microvilli aid in increasing the surface area for reabsorption. So you can actually absorb a lot in that PCT. Now, in the nephron loop, we get both reabsorption and secretion. Now, in the descending limb of the nephron loop, we get reabsorption of water. And then in the ascending limb of the nephron loop, we get reabsorption of sodium and chloride. And we'll talk about how that works coming up in the coming slides. And then in the distal convoluted tubule, what's interesting about this one, you guys, is that the DCT is sensitive to a lot of hormones. So going back to the endocrine system, and we talk about how there's hormones that affect the kidney, typically when we talk about a hormone influencing the kidney, it's going to influence it at the level of the DCT or the collecting duct. So if we talked about how parathyroid hormone helped to increase your blood calcium levels because it allowed you to reabsorb calcium from fluid in your kidney, well, parathyroid hormone would actually influence the DCT to do that. It actually would allow you to reabsorb calcium from your tubular fluid. Or if we talk about how aldosterone aided in potassium secretion, then what aldosterone would do then was actually help remove potassium from blood and then secrete that into your tubular fluid. Or how aldosterone aids in sodium reabsorption. Again, it's going to affect the distal convoluted tubule in that regard. whereas the nephron loop isn't really sensitive to hormones, and it's always going to do the same thing under normal circumstances. Now, the collecting tubule is actually, or collecting duct, is actually sensitive to antidiuretic hormone. So what did antidiuretic hormone aid in the absorption of, or reabsorption of? Water, yeah, exactly. Also called vasopressin, right? It was released during the RAAS, renin-angiotensin-aldosterone system. But antidiuretic hormone released from the posterior portion of your pituitary gland actually helps you reabsorb more water from the collecting duct. So that also helps create highly concentrated urine. So there are hormones that are involved in this process too. So it's actually pretty cool. So let's just go through these parts, you guys. What's the first part of the nephron called?
Renal. Corpuscle. Oh, glomerular capsule. I don't know what you're looking for. There's a lot of structures right there. There's a lot of structures.
Renal corpuscle. Yeah, you're right.
Corpuscle.
The whole thing is the renal corpuscle, you guys. The whole thing is the renal corpuscle. But within that, you got the glomerulus, which is a bundle of capillaries, and the glomerular capsule, which is the outer layer, and then the capsular space. Good. But you're right, the renal corpuscle is the whole thing. The renal corpuscle leads into the proximal convoluted tubule. And what's significant about the PCT?
Has a lot of microvilli.
Lots of microvilli that aid in reabsorption. In fact, most reabsorption occurs immediately in the PCT. And then that leads into a descending limb of nephron loop, which dives down to the medulla, right? That goes into an ascending limb of the nephron loop, comes up out of the medulla, goes to the DCT. What's significant about the distal convoluted tubule? It's sensitive to hormones, right? So certain hormones that affect the kidney often affect it at the level of the DCT, and we'll come back to this in a little bit. And then what's significant about the collecting duct? It's sensitive to antidiuretic hormone. Good, exactly. So is the fluid inside of the nephron considered urine yet? No. In fact, what do we call that fluid inside the urine? Filtrate or tubular fluid. Exactly. And in fact, we don't call it urine until we even get beyond the collecting duct. When's it not technically urine? Out of the papilla when it drips out into the minor calyx. You got it. Because it's not urine yet because it can still be modified. And so modification refers to either reabsorption or secretion. So what this talks about then, you guys, is just basically the different parts of the nephron and what they do, which we just discussed. But if you want, you can pause the slide and read more about it here. Now, earlier I mentioned how the end of the nephron has the distal convoluted tubule, which comes back towards the renal corpuscle, and those things actually can reassociate. So the very end of the nephron associates with the beginning of the nephron, and that we talk about how that forms a juxtaglomerular complex, or JGC, right? This juxtaglomerular complex, its function is to measure different things. Okay, so we got different cells here that aid in the process of measuring, substances and blood pressure and that kind of stuff. So we got macula densa cells, granular cells, and extraglomerular mesangial cells. Now, the macula densa cells you find inside of the ascending limb, right at the juncture of the ascending limb and distal convoluted tubule, right? That's where you find macula densa cells. These macula densa cells, they line the inside of the tubule there. Okay, so they're actually sensing the fluid inside of the distal convoluted tubule. And what they measure inside that fluid is the concentration of sodium and chloride. Okay, that's significant because, and we'll come back to this later, but it's significant that these macula densa cells measure salt inside your tubular fluid, because then you can understand, well, what did your nephron do with that salt? Because we're looking at the end of the nephron, right? If your salt content or sodium chloride content is high, by the time the fluid gets to the end of the nephron, that suggests then that fluid moved too quickly through the nephron, which means you didn't have time to reabsorb substances. Yeah. If the concentration of sodium chloride that filtrate as sensed by the macula densa, if it's too low, that suggests then that your tubular fluid moved through the nephron too slowly, which means you had too much time to reabsorb sodium chloride. So the purpose of this then is the macula densa cells measure sodium and chloride in the filtrate at the level of the distal convoluted tubule to indirectly understand how fast fluid is moving through your nephron, which is weird, but it'll make sense in a little bit when we talk about glomerular filtration rate and how it's modified and changed. That's the function of the macula densa cells. So they sense sodium and chloride in the filtrate. Now, we also have the granular cells. These things surround the afferent and efferent arterioles. And the function of these granular cells is to effectively measure your blood pressure. They measure stretch on the afferent and efferent arteriole. So if your afferent arteriole is really stretched, that suggests your blood pressure is high. Okay? And then what these granular cells can do then is they can contract to prevent damage to your glomerulus. Because if your blood pressure is too high, you could potentially pop your glomeruli. So what these granular cells do then is if they sense too much stretch on the afferent arteriole, they will contract and cause your afferent arteriole to constrict, which also protects the glomerulus. If your blood pressure is too low, like you don't have enough stretch on that afferent arteriole, those granular cells can release a hormone called renin. So if your blood pressure is low, granular cells release renin because they measure blood pressure, right? And renin, as a hormone, is also an enzyme, remember? And so when renin gets in the bloodstream, it converts angiotensinogen into angiotensin 1. Angiotensin 1 was converted to angiotensin 2 by ACE, which is in your lungs, and then angiotensin 2 had 4 main effects, which was to increase blood pressure, right? So it makes you thirsty, causes aldosterone to be released, antidiuretic hormone, and it's a vasoconstrictor. So the function of renin here then, as released by granular cells, is to raise your blood pressure. So when you think granular cells, think blood pressure regulation. Okay. Now we also have these extraglomerular mesangial cells, which are involved with communication between macula densa and granular cells. So they may pass signals between the two. But they're also phagocytes. So these mesangial cells can also engulf debris that gets stuck in the filtration membrane. So macromolecules, like larger proteins and molecules, can get stuck in the filtration membrane. And the way you keep that clean and clear is these mesangial cells can actually clean out that debris, and they actually remove it and keep your filtration membrane clear of debris. It makes sense. Like, let's say if you use a coffee filter once, twice, 3 times, 20 times in a row. Eventually that coffee filter will get plugged, right? It'll get plugged with little tiny bits of material. Even though they're really tiny, you probably can't see them. They'll plug the small spaces in that filter. You know, you wouldn't want that to happen to your body because then you couldn't produce urine. So instead, what these mesangial cells can also do, I know it's not on the slide, but just as a note, they can also remove debris from the filtration membrane. So these mesangial cells keep the filtration membrane clean and clear of debris. So what does this look like then, you guys? Well, what this slide shows then is the renal corpuscle. So all of this is the renal corpuscle. We got our afferent arteriole going into it. We got an efferent arteriole leaving it, and then these are the glomerular capillaries. Okay. The granular cells surround our afferent and efferent arterioles. And then what do these granular cells do if blood pressure is too low? Yeah, they release renin, which acts to increase blood pressure, right? And then what do these granular cells do if blood pressure is too high? They vasoconstrict the afferent arteriole, which actually protects the glomerulus. That's the point of that. It's, you know, if your blood pressure is high, you're not vasoconstricting the afferent arteriole to raise your blood pressure. You're vasoconstricting the afferent arteriole to reduce blood flow to the glomerulus to protect it. Because you can have such high blood pressure, it can pop those glomeruli like little balloons. Just pop, and then that's it. It's done. And then you're going to end up with blood in your urine. So to protect your kidney's glomeruli, then the afferent arteriole can constrict if your blood pressure is too high. That limits blood flow to the glomerulus and therefore prevents, you know, damage from blood pressure. And then what do the macula densa cells do inside the distal convolute tubule? They check salt levels in the fluid, right? And is the distal convolute tubule the beginning or end of the nephron? The end, right? Near the end. So that it's actually measuring salt near the end of the nephron. So it measures sodium chloride levels there. And we'll come back to why that's significant with tuning glomerular filtration rate. But just keep that in mind for now, that macula densa cells sense sodium and chloride in the filtrate. And then what do our mesangial cells here do? They communicate between macula densa cells and granular cells. Good. Good. And they also remove debris from the filtration membrane out here. Very good, you guys. This is what it looks like underneath the microscope. It's pretty amazing. It's amazing to me that we understand the anatomy of this based on this. You know, like people look at these kinds of pictures and can deduce the structure based on, you know, these cross sections, which is pretty amazing.